In 2019, Larry conducted a nationwide survey for beekeeping in Lebanon. Part of this survey included detecting of existing pests and disease in order to generate honeybees pathogen list for Lebanon. Adult honeybees and comb samples were collected based on a defined procedures, and samples were analyzed in Larry laboratories to identify the presence of any potential pathogen. During the microbial analysis, we noticed the existence of several bacteria that were not considered in the list of bacterial disease. And this led to a synthesis that the presence of those bacteria is not accidental, and they might have a role in the immunity system inside the beehive and the bees itself. The scientific curiosity led us to better understand the incidence. Hence, we isolated, purified, and exposed those bacteria to the basic biochemical characterization to be able to define their nature and role. Isolated bacteria were tested against Ascosphaera apis, a special species, Melissococcus plutonus, causal agent of European full brood, and Penibacillus larvae, the causal agent of American full brood, and showed positive results against one or more of the mentioned disease, which validated the beneficial nature of the tested mycelial bacteria. In 2020, with the support of PRIMA program and the Safe AgroBees project, we were able to collect 69 samples of bees and comb from France, Croatia, and Greece. Those samples were subject to same test applied to detect the existence of beneficial mycelial bacteria and isolate them. We succeed in collecting 35 mycelial strains that were subject to the basic biochemical test as well as the antagonistic test against American full brood. The antagonistic test was conducted using the perpendicular strake method in order to select the first 4 strains with the highest antagonistic effect against the American full brood. This test was replicated 4 times, and the mean of the 4 results was considered, and the bacterial strains now are defined. The 4 strains were cultivated to yield big quantity, and the field test was carried on an infected beehive with American full brood. The level of infection was identified through counting of the infected cells in the frames. The test was conducted in an isolated place to prevent the spreading of the disease to neighbor beehives, and the bacteria culture media was sprayed on each frame in the beehive. This application was repeated 3 times with 7 days interval to make sure that during this 3-week cycle, all comb cells have been exposed at least once to the bacterial culture media. In the field, we found prospective results, and we can say that till now 2 infected beehives are still alive. Coming back to the lab, the study continued in defining the type of metabolite excreted from those 4 mycelial bacteria and the time at which the metabolite is secreted. In this perspective, the 4 strains were cultivated in Banat solution, and each 3 days samples were collected, centrifuged to split and concentrate the metabolite. This extracted metabolite was then tested against the American full brood using the well plate technique. Results showed that at day 8, affected metabolites started to be secreted. Those metabolites were subject to enzyme and heat tests to define their nature, which was identified as proteic. At the end, the 4 bacterial strains were sent to Macrogen lab for the bioinformatic analysis to define them. This study will hopefully constitute the basis for revealing a nature potential treatment for the American full brood disease in the beekeeping sector.